autoRAD allows to characterize target radionuclide more effective and rapid than traditional sampling and laboratory analysis.
The main advantage is the separation and online determination using flow scintillation counting fully automated. Thus, it allows the on-site determination of hard to measure radionuclides such as strontium-90.
To begin the system preparation, connect the modular valve positioner exit port to the entry port of a radiation flow detector. Connect a trigger line constructed from a single-board microcontroller to the radio flow detector. Connect the radio flow detector to a container of at least 10 milliliters of scintillator liquid with a high flash point. Place a fresh glass fiber filter in the column channel of the lab on-valve assembly. Ensure that the modular valve positioner is connected both to a waste container and to the LOV assembly. Check the connections of the autosampler and the reagent and waste containers to the LOV. Ensure that sufficient volumes of the liquid reagents are available so that the liquid sampling tubing will remain submerged throughout the experiment. Confirm that the multi-syringe pump is equipped with a 10 milliliter glass syringe and a 3-way syringe valve. Ensure that the in-valve position is connected to ultra-pure water. Connect the out position to the center channel of the LOV via a 10 milliliter PTFE holding coil. Ensure that the autosampler